Afternoon, everyone. It's so good to be with you again. Thanks so much for coming along to learn about the gut microbiome. It really is a fascinating area of research. I must tell you, though, up front, I'm not an expert. I'm not a researcher in this area, just someone who's very interested in the science and where it's headed. It's a vast topic. It's growing day by day. And since I could never do justice to gut microbiome science in the time we have today, I've decided that my talk will cover the basics with a focus on diet. And I hope it's going to inspire you to pursue further learning in this area, depending on the avenues that interest you the most. So let's get started. Right, so all disease begins in the gut. It was over 2,000 years ago that the father of modern medicine supposedly made this proclamation. I'm not 100% sure how accurate that is, but whoever it was, they certainly were very wise. Fast forward to the past 2 decades and to gut microbiome science. It's now apparent that our gut microbiota are critically important for our health and are key regulators of our physiology. Now, microbes have inhabited the earth for hundreds of millions of years, millions of years longer than us. So it makes sense that we would have evolved with them. And I really believe we're living through one of the most exciting periods in medical history. I think the gut microbiome is proving to be the missing puzzle piece that's starting to explain some of the mysteries of human health. It's deeply intertwined with our immune system. It modulates hormonal balance, mood, brain health, the expression of our genes, and our metabolic health, including the health of our cardiovascular system. We're beginning to appreciate that the gut microbiota are actually the command center for human health. So, like I said, this topic is vast and growing exponentially. As you can see from the screenshot, there's been a real explosion of research in this area, especially since 2006 and the advent of DNA sequencing. So since then, there have been around 36,000 papers published, 25,000 of which were just published in the last 5 years, and close to 4,000 were published this year alone. It's mind-boggling. So while in the early years, research was centered mostly around discovering who's there, with as many as 36,000 species of gut microbes identified, in recent years, most of the research is now centered around discovering what they do. But in spite of all the research being done, the science is still in its infancy, and there are more and more questions than answers at this point. So what do we know about the gut microbiota? We know we're living with trillions of microorganisms in a symbiotic relationship. I've heard numbers described as, the equivalent of having 100 galaxies just in our colon. They outnumber our human cells by 10 to 1. These microbes include a complex microbial community of bacteria, fungi, yeasts, viruses, parasites, and archaea. And these are the most ancient of all microorganisms that existed on this planet even before there was oxygen. That's 4.5 billion years ago. And they're inside of us right now. The bacterial population is currently the most well characterized, and the greatest biomass and diversity is in the colon. Microbiota encode over 3 million genes. That's 100 times more than in the human body. The human genome consists only of 23,000 genes, and they produce thousands of metabolites, which we are entirely dependent upon. Everyone has a unique microbiome because we all live and eat differently to everyone else. So no two of us have the same microbiome. I'm going to just show you this diagram just to show you how there is a distinct microbiome in almost every niche of the human body. This will show you some of the main sites, which include the skin and the respiratory system, the urogenital tract. There's an oral microbiome, there's a lot of research going into that at the moment. Our eyes as well have a microbiome, and of course the gastrointestinal tract. And the colon is where the majority of microbial inhabitants will reside. And it's also been the most well-studied historically. The microbial species in the gut have been classified into 11 different phyla, with proteobacteria, firmicutes, actinobacteria, and the bacteroidetes comprising over 90% of the microbiome. So many twin studies have shown that there is a heritable component to gut microbiota, but it's the environmental factors that are actually much larger determinants of microbiota composition. And in fact, these environmental factors start to shape our gut microbiome in the early days of life, and this extends well into adulthood and throughout the lifespan. Now, the gut microbiome has a critical window, and this is considered to be the from conception to the first year of life. And this is a time when it is most vulnerable to environmental influences which can alter its development. Changes during this time can shift the immune system towards a hypersensitive or a hyperinflammatory state, which can lead to a variety of diseases. So I wanted to just focus a little on the main factors that are shaping its development during this critical window. Firstly, the prenatal period. Now, studies have suggested that the introduction of microbes can occur as early as during the prenatal period, with a variety of microbes being present in the placenta, in the umbilical cord, the amniotic fluid, and the meconium of normal pregnancy. And it's believed that the maternal microbiome is likely to translocate into the uterus via the bloodstream, because bifidobacteria and lactobacillus have been identified in the placenta. But it's still pretty controversial at this stage. The role and the function of these microbes in this period is unknown. What is really interesting, though, is that both the mother's vaginal and gut microbiota change during pregnancy. These changes are beneficial. They protect the fetus against certain infections like Neisseria gonorrhea, and they also support species that yield greater energy from the diet, which obviously supports the growth of the fetus. We do know that the first major introduction of a microbial community to a newborn occurs through delivery. As the baby passes through the vaginal canal, it's introduced to the mother's vaginal and fecal microbiome. Now, there are drastic differences between the gut microbiota of a baby born by cesarean section and a baby born by a normal vaginal delivery. Of course, instead of being colonized by vaginal microbes, babies born by cesarean section are going to be colonized by skin microbes. And these microbial differences have been observed as far out as 7 years of age. Now, these deficits in the gut microbiota associated with cesarean section deliveries have been implicated in certain childhood autoimmune conditions, such as celiac disease, type 1 diabetes, and asthma. Not sure if anyone's come across vaginal seeding. Now, this is a process by which the newborn delivered by cesarean section is swabbed all over with vaginal fluids within a couple of minutes after birth. Partial restoration of the microbiota has been reported. However, long-term health effects and the composition of the infant microbiome are not yet known. I've heard that this has been practiced in Australia, but as far as I'm aware, it's not routine in South Africa. Okay, so breastfeeding and formula feeding. Breastfeeding and the introduction of solid food are going to be also major life events that are going to affect the development and the maturation of a newborn's gut microbiome. Now, breast milk can seed the infant gut. It influences the bacteria that follow, and it affects communities right through to adulthood. Breast milk also contains secretory IgA, which is specifically shaped by the mother's microbiota, and so it will protect the newborn from pathogens. Breast milk also contains many important prebiotic compounds, such as these human milk oligosaccharides, which are almost exclusively going to be metabolized by the infant's gut microbiota. They've been shown to promote the growth of key communities and improve barrier function in the infant gut, and obviously inhibit the growth of pathogens as well. So again, well documented, there's a vast difference between the microbiota composition between those exclusively breastfed and formula-fed. Breastfed infants have higher proportions of lactobacilli and bifidobacteria species, a higher diversity and richness even after 1 year. And they appear to be protected against asthma and type 1 diabetes and even obesity. What about early life adverse events? So there is some data from animal studies suggesting that being exposed to early life stress decreases microbial diversity in the gut. In a study of patients with irritable bowel syndrome, those who had gut microbiota distinct from healthy controls were more likely to have a history of early life adverse events and trauma, but these are only associations, and research is ongoing in this area. Antibiotics can play a significant role in or during early life. Several studies have shown how antibiotic exposure can be associated with an increased risk of obesity and diabetes, asthma and allergies, irritable bowel syndrome, and inflammatory bowel disease. There seems to be a delayed maturation of the microbiome as compared to controls, but the exact mechanism by which early antibiotics can predispose children to disease is still unclear. Right, so now let's move on to what our gut microbiome actually does for us. And they really are friends with incredible benefits. So in a return for a place to live and a supply of food, our microbiota work in teams with different skills to perform so many important functions. They have far-reaching metabolic, nutritional, and immunological effects on our health. So firstly, let's look at some of their local functions in the gut. They maintain, they ensure an appropriate intestinal pH, so they can maintain proper functioning of the gut. They ensure proper intestinal peristalsis and a regular rhythm of bowel movements via the enteric nervous system. They also participate in nutrient and energy extraction of food by secreting digestive enzymes, converting complex nutrients into simple organic compounds. They're also involved in fat metabolism, in the breakdown of choline and amino acids, and in the absorption of digested food. They help to increase the absorption of minerals like calcium, iron, magnesium, and phosphorus. They synthesize vitamin K and B12. And gut microbial enzymes also contribute to bile acid metabolism. So what they do is they generate unconjugated and secondary bile acids that then will act as signaling molecules and metabolic regulators. Our gut microorganisms also maintain the structural integrity of the intestinal barrier. They promote mucus production and antimicrobial substances, which all provide protection against the penetration of pathogens and toxins and antigens. And we know, of course, that they are significantly important in the functioning of the immune system. They have modulatory functions. So they regulate the levels of cytokines through the interaction with our gut-associated lymphoid tissue, which is surrounding the digestive tract. Of course, that's also, as you know, considered the largest lymphatic organ in the human body. They regulate intestinal and systemic hormonal function. They modify and eliminate specific drugs. And they also neutralize toxins and carcinogenic compounds. So, gosh, the list is endless. It's probably would be easier to tell you what they don't do. One of the most well-studied metabolites produced by specific gut microbes are these short-chain fatty acids. Now, these are produced through anaerobic fermentation of indigestible carbohydrates. That's mostly fiber. And the main ones are going to be butyrate, propionate, and acetate. So I just wanted to mention some specific functions, both inside and outside the gut, as these short-chain fatty acids have been very well studied. And randomized controlled trials have shown that higher levels of these short-chain fatty acids correlate with a lower diet-induced obesity, with reduced insulin resistance and improved HbA1c levels, which is partly probably via increased glucagon-like peptide 1 production. So butyrate is the primary source of energy for human colonocytes, also very important for stimulating their growth and differentiation. Butyrate induces apoptosis of colon cancer cells. It also is essential for oxygen balance. So it helps epithelial cells to consume large amounts of oxygen. This generates hypoxia, which maintains oxygen balance, and then this prevents the gut microbiota dysbiosis. Butyrate also activates intestinal gluconeogenesis and has beneficial effects on glucose and energy metabolism. And it also regulates osteocyte metabolism, so it has an effect on our bone mass. Butyrate also seems to control gut hormones and can reduce appetite and food intake. That's been seen in animal studies. Propionate is also transferred to the liver, where it regulates gluconeogenesis, and it is involved in signaling satiety through the release of GLP-1. And then acetate, which is the most abundant short-chain fatty acid, this is used in cholesterol metabolism, lipogenesis in peripheral tissues, and it's the only short-chain fatty acid which crosses the blood-brain barrier. So it's thought to be involved with central appetite regulation. So taking all of this into account, it's no doubt that disturbances in the amount and the composition of our gut microbiota are going to lead to numerous abnormalities, such as a disruption of intestinal peristalsis, disturbances in digestion and absorption, disorders in vitamin production or metabolism, difficulties in digesting fats, but also the destruction of the intestinal barrier as well, and excessive stimulation of the immune system will occur. This is actually a really lovely diagrammatic representation of what happens to the lining of the gut when there's this loss of microbial balance, which we refer to as dysbiosis. So when this happens due to damage from various causes, you can see that there's a loss of microbial diversity. And so in the process, a higher proportion of inflammatory microbes will emerge. And if the colon wall is no longer protected by a healthy community of anti-inflammatory microbes, the protective mucus layer and the tight junctions between the cells of the colon wall are going to be damaged, and the wall becomes permeable. Now, bacterial endotoxins like LPS, lipopolysaccharide, as well as food antigens, xenobiotics, inflammatory mediators, etc., they can all move easily through the wall and enter the bloodstream, and wherever they go, they're going to set up inflammation. Lipopolysaccharide is a bacterial endotoxin, which is a component of gram-negative bacteria. And there's also this crosstalk between the microbiota and the immune system. So depending on the species of bacteria, the CD4 T cells, their responses will vary quite significantly. And this can lead to a pro-inflammatory cytokine release, as you can see down there at the bottom on the right. So as you can see in this slide, dysbiosis and bacterial endotoxemia have been linked to a variety of diseases, like autoimmunity, obesity, coronary artery disease, heart failure, type 1 and type 2 diabetes, brain disorders like Parkinson's and Alzheimer's disease, depression, irritable bowel syndrome and inflammatory bowel disease, colon cancer as well, other cancers like lung, pancreatic, breast cancer as well. What else? Pancreatitis, metabolic syndrome, non-alcoholic fatty liver. So dysbiosis it really seems to be at the root of most modern diseases. What are some of the common things that actually damage the gut microbiome? Well, diets, particularly a diet that's high, or an ultra-processed diet that's going to be high in fat and sugar, this is going to feed the unhealthy inflammatory microbes, which are going to in turn produce unhealthy metabolites. Study after study show that high-fat diets cause an unhealthy balance and impair the barrier function of the gut. Now, high animal protein diets as well can increase inflammatory microbes, and these can produce more toxins, amines, sulfides, which are associated with ulcerative colitis, and secondary bile acids, which have been associated with colon cancer. And then you also get substances like L-carnitine and choline, which are converted into metabolites called trimethylamine. This is then converted to TMAO in the liver, and this is known to increase the risk of coronary artery disease and stroke and Alzheimer's as well. Then we have high-intensity sweeteners. These have also been shown to cause dysbiosis and insulin resistance and liver injury as well. Food additives and emulsifiers, they reduce microbial diversity, they induce inflammation, they can promote obesity and colitis and colon cancer. Highly restricted diets, so diets like the low FODMAPs diet, gluten-free, the paleo, the carnivore diet. These diets can cause dysbiosis, and it's because every dietary choice that you make, it empowers a specific group of microbes. And so if you permanently remove a food group, the microbes that thrive on that food are going to starve into extinction. And then alcohol is another component which causes dysbiosis. Other common causes include disruption of circadian rhythms, stress, a lack of physical activity, chemicals like herbicides and pesticides, smoking, and then drugs, many drugs, many more than I've listed here, but the most important ones are going to be your proton pump inhibitors, These increase the risk of small intestinal bacterial overgrowth, and antibiotics, of course. A few days of treatment are going to wipe out about a third of your gut microbiome or gut microbiota. Most people will recover in about 4 weeks, but it can take as long as 6 months, and in some cases, it's been reported that recovery can take a few years. Other drugs include osmotic laxatives and oral contraceptives. Non-steroidal anti-inflammatories can also be the microbiota that destroy the lining, of course, and can create ulcers, predisposing to microscopic colitis and inflammatory bowel disease. And then we have the TNF alpha inhibitors used to treat rheumatoid arthritis. So as you will appreciate, lifestyle factors strongly impact the composition of the gut microbiome. I wish there was more time today to go into some of the mechanisms, but we'll have to leave that for another time. But there is so much that we can do that's within our control. So of all the pillars of lifestyle medicine, the nutrition pillar is really one of the most powerful interventions for improving the gut microbiome. So I'm going to focus on diet. So let's take a deeper look at that. So as well as the diet, of course, having a direct effect on our physiology and health, as I've said, there's growing evidence now that our diet may also have an indirect effect on our health by altering our gut microbiota composition and the production of metabolites. And this is a similar diagram to the one before, but I think it shows really clearly the effect of diet on the gut microbiome. And so just to reiterate again, as you can see on the left, you've got a healthy diet, which is going to be high in fiber and polyphenols and omega-3 fatty acids. And this is going to contribute to the production of short-chain fatty acids, of course, which are going to enhance mucus secretion and support the healthy bacteria to produce antimicrobial peptides. They also support the health of the colonocytes so that there's an increased expression of tight junction proteins. And then the short-chain fatty acids are going to reduce oxygen levels, and they're going to maintain a functional immune system. But what happens when you have, when the diet becomes Western, when it becomes full of ultra-processed foods? Now you have low fiber, you've got high protein, high sugar consumption, high fat. The diversity of your gut bacteria are going to be reduced. Their function is altered. So now you've got toxic metabolites being formed and toxic bile acids. And these are going to increase one's susceptibility to infections, to inflammatory bowel disease, stimulating a pro-inflammatory immune response, and then eventually chronic inflammatory diseases. But what is the evidence for all of this? So I've chosen just a few studies to provide you with some evidence for these changes. This is a study from 2016. Research sequenced the gut microbiomes from 1,135 participants from a Dutch population cohort. And they looked at food frequency questionnaires and microbiota type and determined the association between 63 dietary factors and microbiota composition. And the results were that a diet higher in ultra-processed food produced microbiota that were less dense and diverse, and they were producing harmful metabolites. And diets high in fiber-rich foods, fruits, vegetables, whole grains, the microbiota were much more diverse, they were dense, and they produced much more short-chain fatty acids. Another study, much closer to home, a really interesting study, was a collaboration between researchers from KwaZulu-Natal and the US, and they explored whether colon cancer risk is determined by the influence of the diet on the microbiota to produce anti or pro-neoplastic metabolites. So there were two groups. They performed a series of investigations between a group of African Americans and a group of rural South Africans. And the animal protein and fat intake was 2 to 3 times higher in the Americans, whereas carbohydrate and fiber, chiefly in the form of resistant starch, were much higher in the South Africans. And on colonoscopy, the African Americans had more polyps and they had higher rates of mucosal proliferation. And these differences were shown to be associated with the microbiota. Now, the American microbiota were also dominated by higher levels of potentially pathogenic proteobacteria and bile acid deconjugators, which means they were producing more secondary bile acids that we know are carcinogenic. Whereas the South Africans, their microbiota were dominated by Prevotella, which are butyrate producers, and butyrate is an anti-inflammatory and anti-neoplastic. So secondly, the researchers then went on to subject the groups to 2-week food exchanges, where the African Americans were then fed a high-fiber, low-fat African-style diet. I think they increased the fiber from 14 to 55 grams a day. And the rural Africans, they were given a high-fat, low-fiber Western-style diet. And this was all under close supervision for 2 weeks. And so in comparison with their usual diets, the food changes resulted in remarkable changes in mucosal biomarkers of cancer risk. In the African Americans, there was increased fermentation and increased production of butyrate and a suppression of secondary bile acid synthesis. So it looks like if you want a healthier microbiome, you really need to increase your intake of fiber. But just in case I haven't convinced you, I want to share one last paper with you. This was a paper by Reynolds, published in The Lancet in 2019. And this really is the highest quality study that's been done to date that examines the effects of dietary fiber on human health. So what they did is they put together 185 prospective cohort studies, 58 randomized control trials, and this encompassed a phenomenal 135 million person years of data. And they found, so just looking at these forest plots here, you can see it's quite obvious that all of those outcomes were positively associated with a higher fiber intake, particularly systolic blood pressure and HbA1c and body weight. And this was both for total dietary fiber and for whole grains. What were some of the other results? Well, those who consumed more fiber, they had a reduction in all-cause mortality. They also had a reduced risk of coronary heart disease. They reduced risk of type 2 diabetes. They were less likely to be diagnosed with colon cancer and breast cancer and esophageal cancer as well, and reduced mortality from cancer. They also were reported to have a reduction in stroke, more likely to lose weight, to reduce blood pressure and cholesterol levels. And the protection was proportional to the amount of fiber consumed. So how much fiber are people actually consuming? So according to the World Health Organization, adults should eat at least 25 grams of fiber per day. while the dose response data suggests that amounts greater than 30 grams per day confer additional benefits. And in fact, recent suggestions are that we really should be getting up to about 50 grams a day if we want to achieve health benefits. And remember, our ancestors, probably before the time of agriculture, were consuming more than 100 grams a day. But the reality, as you can see from this diagram, is that most people are not getting, nearly enough. And in fact, 97% of people around the world are fiber deficient. You can see here that most people around the world are consuming less than 20 grams of dietary fiber per day. And even in South Africa, our average is only 21 grams, according to this table. In some areas, it's so low. I found one study that was conducted in Ellisras in Limpopo province, and they looked at the consumption of dietary fiber in 627 individuals between the ages of 18 and 30, and they found that the average total fiber intake was only 4.6 grams per day. So we really have a huge amount of work to do if we're going to reduce non-communicable diseases through diet. Now, achieving the high fiber intake is challenging. It's probably only going to be achieved by supplementing the food supply with dietary fiber sources. And there's also the problem, of course, that modern humans are most likely going to have problems tolerating such high fiber doses, at least initially. High fiber diets can lead to flatulence and bloating and pain, even diarrhea and constipation in people who don't have healthy microbiomes. So the tolerance is really dependent on the individual, but it does improve over time as the gut and the microbiota adapt to the higher doses. If people don't respond at all, it could be that they're missing some keystone species and strains that would be, normally utilizing specific fiber sources. And so it's been suggested that in the future, we might even be able to match people with, so look at their microbiota profiles and then actually match those profiles with specific fiber types that could be used in a very personalized way. But this is still very much in the future. And so right now, to reduce side effects, you can advise your patients to increase their fiber content slowly in small increments as they tolerate it. So for the remainder of the talk, I would like to focus on prebiotics and probiotics. The probiotic supplement market is, I think, worth in the region of $70 billion, and it's expected only to increase between now and 2030. So what evidence-based advice can we give patients about prebiotics and probiotics? Just to start off with, and so for some clarity on terminology, so the prebiotics is your fiber, or the not only fiber, but the food for your microbiota. And symbiotics have been defined as a mixture comprising both the live microorganisms and the substrates that are selectively utilized by the host microorganisms, and that they confer health benefits on the host. What are postbiotics? So postbiotics is defined as a preparation of inanimate microorganisms, so they're not alive, or the components that confer a health benefit to the host. So they must contain either inactivated microbial cells or cell components, with or without metabolites. These definitions come from the International Scientific Association of Prebiotics and Probiotics. And I really recommend that you take a look at their website. The address is down there at the bottom of the slide. They have developed a series of some really good scientifically based, patient-friendly resources, which you could use in your practice. This is an example of one of their infographics that helps to explain key concepts that are related to prebiotics and probiotics and fermented foods. So this one is on fiber and prebiotics. So talking about fiber has been classically classified based on solubility and fermentability. And soluble fiber dissolves in water and can be fermented in the colon. And insoluble fiber doesn't dissolve, but it adds bulk to stool, and it's less likely to be fermented. There are, however, some types of insoluble fiber, like resistant starch, for instance, which can be fermented by gut microbes in the colon. And so then it would be considered a prebiotic. So prebiotics are substrates that are selectively utilized by the host microorganisms and confer a health benefit to the host. And here are a few food sources. So resistant starch, as I mentioned, found in barley and millet, legumes, cooked and cooled pasta, rice and potatoes. Then we have soluble fiber that's found in legumes and oats and barley, in the flesh of fruit and vegetables. We have oligofructose found in garlic and onions, wheat and legumes, and then galacto-oligosaccharides also found in wheat and legumes. These are the two that are going to cause issues with people, for people with irritable bowel syndrome. And then inulin as well, found in chicory root, leek, asparagus, and banana. And then the polyphenols, which are your plant pigments. These are also used by microorganisms, and so would also be considered prebiotics. What about probiotics? So probiotics are the live microorganisms that, when administered in adequate amounts, confer health benefits to the host. Most commonly, or the most commonly used probiotics comprise the lactic acid producing bacteria. And if they're in food, they must contain at least 1 million of colony forming units per gram of viable and active microorganisms, whereas freeze-dried supplements have shown good results with, I think that's 10 million to 100 billion viable microorganisms per day. The properties of probiotics also are very species and variant specific. There's also this common misperception that for a probiotic to be effective, it has to impact the composition of your gut microbiota. But this is not true. Probiotics typically do not take up residence, so they do not colonize the gut, and they may not actually result in any detectable changes in the microbes that are normally present. But as they pass through the gut, they interact with our immune cells, our colonocytes, the dietary components in the gut, and the microbes that are living in the gut. And so that's how they exert their benefits. Now, we can also find probiotics in fermented foods or live bacteria in fermented foods. We wouldn't necessarily call them probiotics. Fermented foods include miso, sauerkraut, kimchi, pickles, kombucha, kefir, and yogurt. So fermented foods, as I said, are not necessarily probiotic foods. Only some fermented foods contain microbes that meet the strict criteria to be called a probiotic. So that would usually be your yogurts. Probiotics can also be added to nutritional supplements or to foods that are not fermented, such as fruit juice or cereal bars. Let's see what else. Oh, yes. Now, there's been a lot of extensive research into the differences between various probiotic strains and how they interact with our immune system and regulate our metabolism, etc. Affect our brain health or our gut neuromuscular function. But in spite of all of these randomized trials and clinical data, we don't have comprehensive guidelines on the evidence for various probiotics. And so this is probably due to the fact that there's so many other factors that are influencing the effectiveness of a probiotic in any individual. These probiotics need to compete with pathogens in the gut, for example, Helicobacter pylori. And there's also the unique background of the individual, such as genetic factors, baseline immune functions, that person's particular microbiome, that person's, the environmental factors such as diet and stress that are going to be affecting that unique individual. So this is probably why there really isn't any conclusive evidence at the moment. So I would recommend that if you do want to use probiotics, that you choose a strain or a blend of strains that are backed by science supporting the benefit that you are looking for. And this is a really good online resource. It's US-based, but you are able to look up specific conditions, and then you can check on the exact probiotic strains or the variants that are recommended based on different levels of evidence. So, in summary, targeting the composition and metabolic functions of the gut microbiota is certainly a really, really good option for the prevention and treatment of disease, but there are still so many unanswered questions. And I just wanted to leave you with some of the questions that we have, and this is really going to be where future research is focused. We still don't really understand what is a good gut microbiome. How can we diagnose intestinal microbial disorders clinically and develop reliable screening methods for accurate diagnosis? Is the intestinal microbial disorder the cause or the result of the disease? In many cases, we're not sure. Is the disease due to a specific strain of bacteria, or is it the overall intestinal microbial composition? What are the exact mechanisms of disease, if they are caused by the gut microbiota? Still trying to determine that. And then, if those microbes are found to be beneficial in the treatment of disease, can they also prevent disease? So still so many unanswered questions. But in the meantime, I think one can focus on what we do know. There are things we do know that improve the health of our gut microbiome. And we can improve its diversity, its stability, and its resilience. And so this is what I'm doing and advising my patients. And so I'm going to leave you with these 7 tips. Eat a whole food, diverse, plant-based diet. Aim for more than 30 different plant foods a week. This is data that came from the American Gut Project. They specifically found an advantage when 30 or more different plant foods were consumed each week. Aim to consume more than 30 grams of fiber per day at least. Eat the rainbow, so increase your polyphenol intake. Eat regular servings of fermented foods. Avoid ultra-processed food. Practice time-restricted eating. 10 to 12 hours overnight is probably fine. Remember the circadian rhythms that I mentioned that can have a negative effect, or a problem with circadian rhythms that can have a negative effect on the microbiota. And then practice stress reduction techniques and keep physically active. And that brings my presentation to a close. Thank you so much for listening.